This is Krista Zanetti. I would like to thank you for participating in the NIH Metabolomics Interest Group webinar series. We're very excited today to have the webinar from Dr. Christina Meni that will focus on metabolomics of blood pressure regulation. Before introducing our speaker, I would just like to quickly review a few logistics. And where do I change from? Sorry about that. Okay, should you experience technical difficulties, you can contact us through the questions box in the expanded control panel or by phone or by email. If you need to view live closed captioning, please click on the link that will appear in the chat box. At the end of Dr. Meni's presentation, we will allow time for questions. If at any time during the presentation you have a question, please type it into the question box, and I will ask it on your behalf during the Q&A session. I'm going to go back. So it is my pleasure to introduce Dr. Christina Meni. Dr. Meni is a research fellow in the Twin Research and Genetic Epidemiology Department at King's College London. She received her Master in Mathematics from the University of Cambridge and her PhD in Statistics from the University of Milan Bicocca, where she specialized in statistical genetics. Dr. Meni's research is focused on identifying novel molecular markers associated with aging and age-related diseases such as hypertension using metabolomics in conjunction with several other omics data sets. She has co-authored over 40 scientific articles in peer-reviewed journals. She is involved in the data quality control, analysis, and interpretation for various European Union and Medical Research Council projects within King's College London. As I mentioned, Dr. Meni will be presenting on the metabolomics of blood pressure regulation, and we'd like to thank her for joining us today and welcome her for her presentation.
Hi, thanks Krista for the introduction. So I'm going to talk about metabolomics of blood pressure regulation. I will first give a little introduction on hypertension, and then we'll present a metabolomic study of blood pressure, some pathway analysis, and then a metabolomic studies for arterial stiffness and some microbiome analysis. Hypertension is the leading risk factor for cardiovascular for morbidity worldwide, with an annual death rate of about 7 million people. And it has a prevalence of 30%, with about 1 billion people with hypertension worldwide, and this is expected to rise to 1.5 billion by 2020. What is hypertension? Hypertension is high blood pressure, and blood pressure is the pressure of blood in the arteries. We can measure systolic blood pressure, which is the pressure of the heart when the heart beats, and diastolic blood pressure is the pressure in the arteries when the heart releases. And hypertension is defined as a blood pressure higher than 140/90 millimeters of mercury. In this picture, you can see the risk of cardiovascular death according to baseline blood pressure in men. You have systolic blood pressure in the bottom and diastolic blood pressure on the right. And as you can see, the relative risk is significantly higher in people with high systolic and diastolic blood pressure. And here it's plotted the risk of myocardial infarction in yellow and of stroke in red in 5 years for normotensive versus hypertensive individuals. And the risk, as you can see, is much higher in those who have hypertension. So hypertension is associated with 69% of first myocardial infarction, 74% of cases of coronary heart disease, 77% of first strokes, and 91% of cases of heart failure. And is therefore a very big public health burden. However, blood in a big meta-analysis of 1 million individuals, it's been shown that a reduction of 2 millimeters of mercury in systolic blood pressure reduces the risk of mortality for ischemic heart diseases of 7%, and also reduces the risk of mortality from stroke of 10%. It is therefore very important to control blood pressure levels, and at the moment hypertension treatment follows a trial and error approach, with only a third of hypertensive patients actually achieving target blood pressure. So it's very important to identify markers, early markers of people at risk of developing hypertension, and also markers that enable treatment of stratification. This is the current landscape of blood pressure genetics now. As you can see, there are many genes involved in blood pressure regulation, and however, all this collectively only explain 0.9% of the phenotypic variance. And so we need other ways of explaining the remaining of the variance. And Metabolomics is a new high-throughput technology which measures endogenous metabolites in cells, tissues, and other biosamples. And this metabolite capture a snapshot of the metabolic profile of the individual and allow for the potential identification of early biomarkers. Metabolites are intermediate phenotypes, and they can capture both intrinsic and extrinsic factors. They've got very dynamic nature, which makes them ideal for measuring physiological response to external stimuli or the development of pathogenic processes involved in cardiovascular diseases. So our aim was to discover novel molecular markers associated with blood pressure regulation and treatment using the twin-CK cohort. The twin-CK register was started in 1992, and it now contains over 13,000 twins, mainly females, with a very wide age range. We have almost equal number of identical and non-identical twins, and we have over 1,000 phenotypes collected, and also longitudinal data available. Each twin completes a couple of questionnaires per year, which includes medical history and lifetime factors. And we also have a high percentage of twins invited to the hospital for more detailed phenotyping and sample collection. Besides phenotypes, we also have cutting-edge molecular phenotypes like GWAS data on about 7,000, expression data in different tissues, epigenetics data, and metabolomics data on over 6,000 measured with different platforms at different time points. And so altogether, these omics can be combined in order to discover pathways. So now I'm going to talk about our metabolomic study of blood pressure. So for this, we included 3,900 female twins from Twins UK who did not have renal impairment and were not on any antihypertensive treatment. And we analyzed 280 metabolites measured with the Metabolome platform. We then did survival analysis on the significant independent metabolites. We replicated our results in two independent populations, one from Germany, Cora, and the other one from England. And then we did some animal models to assess causality. So these are the results for systolic blood pressure after adjusting for covariates, multiple testing using Bonferroni. And as you can see, the majority of the metabolites are lipids that have been color-coded in blue. But we also identified quite a lot of amino acids, 4 carbohydrates, some peptides, xenobiotics, and metabolites involved in the energy pathway. And these are the results for diastolic blood pressure, which are very similar from the one for systolic blood pressure. These are 63 metabolites, and they all overlap with those associated with systolic blood pressure. So then we did some data reduction and identified a panel of 15 metabolites that were independently associated with the blood pressure. And among those, the decarboxylic acid hexadecanedioate was particularly interesting because not only was associated with both systolic and diastolic blood pressure, but was also associated with all-cause mortality. And we replicated these results in Cora and Hertfordshire. So these are on the right-hand side. You can see the survival analysis for hexadecanedioate, and we find that circulating levels of hexadecanedioate are consistently associated with higher risk of mortality. So then, in order to determine whether hexadecanedioate was causally associated with systolic blood pressure and it was not a byproduct of blood pressure elevation, we performed some animal experiments. So in panel A and B, we used salt-resistant Kyoto rats, and we fed them hexadecanedioate in their diet. So the hexadecanedioate treated rats are those in white, and as you can see, rats fed hexadecanedioate levels had significantly higher systolic blood pressure. We then wanted to show reverse causality, so we used spontaneously hypertensive prone rats, and we fed them salt. The cases are the one taking salt are the one in white, and so after salt intake, the rat had higher systolic blood pressure. However, having a higher systolic blood pressure did not cause a higher hexadecanedioate level. We then repeated the first experiment measuring systolic blood pressure via radiotelemetry, and after a 7-week recovery period, you can see that the rats taking the metabolite in their diet, the one in red, had significantly higher systolic blood pressure compared to the control. So what we see is that hexadecanedioate is causally associated with blood pressure. Hexadecanedioate is a byproduct of omega oxidation of fatty acids, which is a minor pathway for fatty acid oxidation, which is used when beta oxidation is deficient or overwhelmed. However, we don't know how this affects blood pressure. So then we use our Metabolomics to look for molecular pathways which could potentially influence the metabolite levels. So our first question was whether we could find genetic variants associated to the level of the metabolite using GWAS data and transcriptomic data, and to see whether those variants were then linked to hypertension. So we look up the top association for hexadecanedioate in a genome-wide association scan, which we performed previously in collaboration with Cora. And here you can see the Manhattan plot for hexadecanedioate, and on the right hand, well, you have on the x-axis the chromosome position and the y-axis minus log 10 of the p-value. As you can see, there are 2 strong signals, especially 1 strong signal on chromosome 12. So we find 242 SNPs associated with circulating levels of hexadecanedioate achieving GWAS significance. And all these SNPs map to 2 genes. The one on chromosome 12 is the solid carrier and organic anion transporter, and on chromosome 1 is a cluster of Cytochrome 4. So SLCO1B1 on chromosome 12, this is the locus plot. So again, on the x-axis you have the chromosomal position, and on the y-axis the minus log 10 of the p-value, and the top SNPs are holding high linkage to equilibrium. SLCO1B1 encodes a liver-specific member of the organic anion transporter family, which is involved in the pharmacogenetics of statins. And we then go on to look at the association between SLCO1B1 and hypertension. And in Caucasian, we find the hexadecanedioate increasing allele of the top SNP has been associated with statin myopathy. However, we find no association with blood pressure or hypertension. We looked at two GWASs, the ICBP consortium GWAS, which includes over 65,000 people, and also a case control GWAS for extreme of blood pressure. We did find one variant of SLCO1B1 associated with hypertension in the extreme GWAS, but the p-value was quite small. We then looked for other ethnicity, and we found a study in which an African American from ARIC, where a loss of function variant in SLCO1B1 leads to high hexadecanedioate levels, and the same allele was also associated with risk of heart failure and with statin-associated myopathy. And in the Uyghurs, in some Asian population, genetic variation in the promoter of LCL01B1 gene has been implicated in risk of essential hypertension. We could not test, however, this variant in our data because it's monomorphic in Caucasians. We decided also to look at for association between hexadecanedioate and MI, starting from the result that was found in African Americans. And in our sample, we find a consistent role of this compound in cardiovascular risk, with a significant higher odds ratio for people who have high hexadecanedioate levels. However, our sample size was quite small. This is the second strongest gene to be statistically associated with hexadecanedioate, and is the Cytochrome 4 cluster on chromosome 1. So again, in the locus plot, there's a high LD. And this gene is very interesting because it's involved in the omega oxidation of fatty acids. So hexadecanedioate accounts for the omega oxidation, and in the first step of the omega oxidation there is the introduction of a hydroxylic group onto the omega carbon, and the oxygen for the group comes from a complex reaction which are conducted by members of the CYP4 family. And moreover, we find that expression levels in fat, but not in blood, of CYP4 are positively correlated with hexadecanedioate in blood. CYP4A11, which is one of the genes in the cluster, is responsible for metabolizing the arachidonic acid 28, which is a metabolite which is involved in the regulation of blood pressure. And again, we did not find any association between SNPs of the CYP4 genes and hypertension in the two GWASs on Caucasian, but polymorphisms on this gene are involved in essential hypertension, both in the Chinese population and in African Americans. And in the kidneys, the 28 contributes to antihypertensive mechanisms, and it also promotes endothelial dysfunction in the microvasculature. And it's involved in the renin-angiotensin and aldosterone system. We then went to look at our transcriptomic data to see if we could find any biological relevance of hexadecanedioate. And we looked for, we had 74 individuals which were included in the analysis. And these are the top results. The top gene is the alkyl dehydrogenase gene. And this is again very interesting because alkyl dehydrogenase is involved in the second step of the beta omega oxidation. And this is the same picture as I showed you before. So here we would have CYP4 and then second step alkyl dehydrogenase. And moreover, we also find significant association between circulating levels of hexadecanedioate and 3 SNPs on the ADH1B gene, suggesting that the gene is actually implicated in hexadecanedioate regulation. So again, we find no association between these SNPs and hypertension in Caucasian, but a role of ADH1B has been found, and hypertension has been found in Japanese men, in particular in relation with alcohol-induced hypertension. So then we explore the relationship between alcohol intake, hexadecanedioate, and blood pressure on our data. And we find that, so in the picture you find hexadecanedioate divided into tertiles, and in pink individuals who have just 1 standard drink per day, and the one in dark red people consuming high lots of alcohol. And we find that alcohol intake has no effect on systolic blood pressure for people with low hexadecanedioate, and there is a significant difference in people with high hexadecanedioate, suggesting that this compound might have a role in alcohol-induced hypertension. So in summary, we find that circulating levels of hexadecanedioate are associated with variants that result in hepatic uptake of organic anions and to genes encoding enzymes involved in the omega oxidation. And all these genes have been associated with hypertension, even though we find no association in Caucasians. And so further studies are needed to establish mechanistic basis of blood pressure regulation via hexadecanedioate. And this is just a picture showing you some of the pathway that we have identified. So we have high hexadecanedioate causing high blood pressure. We have low uptake and reuptake of hexadecanedioate via SSCO1B1, and then the genes involved in the omega oxidation of fatty acids. And now I'm just going to briefly talk about another study we've done on metabolomics and pulse rate velocity. So pulse rate velocity is a measure of arterial stiffness, and arterial stiffness is the most important cause of increasing systolic blood pressure beyond the age of 40. And it's also been shown to be an independent predictor of cardiovascular disease and mortality in middle-aged and older adults. So again, we included 280 non-metabolites from the Metabolome platform and 1,700 female twins who were not on antihypertensive medication. And the metabolites that were associated with pulse rate velocity were then entered into a backward linear regression. And after a stepwise regression, pulse rate velocity was significantly associated with 3 metabolites: uridine, serine, and phenylacetylglutamine. And then we tested for correlation with traditional risk factors like Framingham. So here on the left-hand side panel, you find the association of the 3 metabolites with pulse rate velocity. And on the 3 pictures on the right, you see the association between the 3 metabolites and the Framingham risk score. And as you can see, uridine and serine are associated only to pulse rate velocity, but not with the traditional cardiovascular risk factors, while phenylacetylglutamine is consistently associated with both pulse rate velocity and the traditional Framingham risk factors. So uridine and serine are not related to traditional risk factors in our samples. Uridine is known to be an antagonist of the P2 receptors, which are the purinergic receptors involving eNOS phosphorylation during endothelial activation. In the twin CK samples, uridine levels were nominally associated with gene expression levels in fat of P2Y2, and this suggests that it may play a role in regulating E0 phosphorylation, which may then influence arterial stiffness. Phenylacetylglutamine was particularly interesting because it's associated with blood pressure, with the Framingham risk score, and with pulse rate velocity, and is also strongly associated, and this part goes Ferroni, with gene expression level of the CDEC gene, which is a gene related to insulin resistance, and also is the metabolite is related to the gut microbiome composition, and a number of reports have linked the function of gut bacteria to insulin resistance. So the gene CDEC may be linking these two pathways and suggest a new avenue of research regarding the role of insulin resistance and the gut microbiome in blood pressure regulation. So these pictures come from Metabolomics, and these are all the 280 metabolites that we have measured. And in red, the metabolites are exclusively or mainly contributed by bacteria metabolism. And phenylacetylglutamine comes from the gut microbiota. And so we decided to look at an association between phenylacetylglutamine and microbiome in our own data. And we found lots of significant hits. And these are just some of the top. And the bacteria related to phenylacetylglutamine come mainly from two families, the Christensenella and the Ruminococcus. Christensenella is associated with leanness in humans and prevents weight loss on the high-fat diet in mice. And since phenylacetylglutamine is associated with visceral fat, insulin resistance, and blood pressure, some of the effects of this bacteria may be mediated via phenylacetylglutamine. Ruminococcus family, the bacteria in this genus are specialist primary degraders within the microbial community, and they are able to release energy from particular substrates such as dietary fiber and resistant starch in the human colon. So in conclusion, using metabolomic profiling, we have identified novel pathways related to blood pressure regulation. We show that an integration of metabolomics, transcriptomics, and genomics has the potential to unravel novel blood pressure pathways that have potential use for clinical translation, early diagnosis, and treatment stratification for hypertension. Thank you.
Okay, thank you so much. Right now, we're going to take some questions. So do we have any questions from the viewers? You can just type them right into the question box. So I'll give you a minute to do that. Oh. Okay, I will ask the first question while the rest of you are able to think about the questions you want to ask and have some time to type them in. So, so Christina, I was wondering, so the twins UK is 85% female. So going back to sort of the sample size you used, I'm just curious about this. So that's about a little over 11,000 that are female. So I understand why you had to why you chose females only for your studies. But I'm wondering a couple of things. One, your results, how generalizable do you think they are to a male population? And two, I was wondering when you replicated or validated or replicated your results for the hypertension in the Cora and Hertfordshire studies, did those include females only, or did you attempt to sort of generalize those to men at all?
Yes, so the both Cora and Hertfordshire have both men and women, so I think our results are quite generalizable. And for this particular analysis, we excluded men because there were not that many which had the phenotype, were not on blood pressure medication, and didn't have renal impairment. But the other cohorts, it replicated well both with men and women.
About what do you remember? What sample sizes you replicated in Core and Hertfordshire?
The percentage of men.
No, I'm just curious of the total sample size, and I guess and the percentage of men. I'm just curious what you used for replication.
So there were 1,500 roughly in both.
Oh, great. Okay. And do you recall the percentage of men?
Yeah, 50%, something like 50%.
Great. Yeah, I was just curious about that. I think because obviously I think I calculated it really quickly, and really in the Twins UK you have just shy of 2,000 men. So I think it'd be really hard to actually get the numbers you needed to do the study. So I think it's really good to hear that you did look at this to see if it was generalizable to men. So thank you for answering that. We did get a few questions. I'm going to start reading them. So the first one is, you have a new paper in PLOS One on the pharmacogenetic footprint of ACE inhibition. Could you say a few words on that?
What about? Like anything in particular?
Okay, we're going to give the person who typed it in just a second to elaborate on that, if he would like.
Yes, thanks.
I'm going to, Karsten, we'll give you a few minutes to elaborate, and I'm going to move on to the next question. And then if you have something more specific that you'd like her to comment on, we'll come back to that. Okay, and the next question is from Steve Moore. He said, great talk. What do you think are the major sources of variance in the I'm not even able to say it, the hexadecanedioate, is it from the diet or is it genetic?
Well, I think it's a little bit of both. It has an okay heritability, and we find good, well, very good hits genetically, but there is definitely a contribution to diet, and is associated, for example, with the Mediterranean diet. And so there could be a way of controlling the hexadecanedioate level by changing your diet, which is something we are looking at the moment. If that answers the question.
Great. Thank you. Okay. So we're going to follow up with the first question from Karsten Schur. And Karsten, I hope I'm pronouncing your last name correctly. So he he said that was a mean question, and now I called out who it was. I'm involved in that one, and was just wondering whether Dr. Meni had some backup slides on that one that she would like to, and whether she'd like to talk about it.
No, I don't. Sorry.
Okay. Well, we can always invite you back to talk about that one. So just if you do have any comments on that, Christina, as we move on, you can just chime in. Okay. We're going to move on to the next question from Andrew Monty. Have you begun to look at any treatment effects of these hypertensive patients? For instance, do you see changes in the hexadecanedioate levels in patients that start antihypertensive treatment? Did we lose you, Christina?
Yeah.
Did we lose her? Okay. We're going to have to just calling back in.
Yeah.
We're going to have to just give it 1 minute. We just lost her. She's coming back on. She's coming back on in 1 minute, so please just give her, we have several questions. She's jumping back on, and she will answer those. Give us one second.
Hello. Hi.
Hi, you're back. We realized we lost you. I'm going to read. That's okay. It happens. You were very quick jumping back on. We are going to read. I'm going to read that question from Antrumonti again. Have you begun to look at any treatment effects in these hypertensive patients? For instance, do you see changes in the hexadecanedioate levels in patients that start antihypertensive treatment?
No, the answer is no. We haven't, because we don't have enough people on antihypertensive treatment. So we've done a little bit on the drugs, like as Carsten was saying with ACE drugs, but we don't have enough information in order to do some more detailed analysis.
Okay.
But we are in a MSc grant, which is looking at drugs and things.
So there's more to come.
Yes.
Great. Thank you. The next question from Joe Lee is, were the two cohorts assayed using the same platform?
Cora, yes. The other one, no. It was done using a targeted approach in the lab. So they developed the metabolite just for us.
So it was Metabolon for the large hypertension study, and then Cora and Hertfordshire were on targeted platforms.
Yes. Yes.
Okay, great. Thank you. Next question. Also for the hexadecanedioate, was the association with mortality driven by blood pressure or independent?
The association with mortality was there even after adjusting blood pressure. So I'm supposing it's independent.
Okay. And another question, were there significant metabolites that were specific to one sex but not the other?
Well, in general or in this analysis?
I think I'm going to assume in this analysis, but if the asker would like to follow up, we can take a follow-up question if I'm interpreting his question incorrectly.
Okay. No, so in this analysis, we all included females because there were only about 200 men who met the criteria. So we didn't do the analysis stratifying by sex, and in 200, so the results were quite consistent overall and in the female samples. However, in the male sample, they were too little to be significant, or we didn't look into it too much.
Okay, so great. So I just want to check. Joe, did she answer your question? Because I got two follow-ups, and I want to make sure whether or not she answered it. Could you just type yes or no? And then if not, I'll follow up with your other questions. Okay, so we don't have. Okay, so Joe, I'm going to assume she answered your question unless I hear from you a follow-up. The next question is, among your 15 metabolites associated with blood pressure, were there any other interesting targets? Also, what was the direction of the effect for coffee metabolites, the paraxanthine?
So coffee was associated actually with less mortality, caffeine. So it was also, so it was protective, and it came up in the 15 metabolites. And however, coffee intake was associated with higher levels of blood pressure, but then it was also associated with lower mortality. So the direction was not consistent.
Great. Okay, we do have another question. The next question is, were there any changes of these blood pressure-related metabolites with aging?
Yes, lots of them, but all the analysis is adjusted by age, so whatever we are seeing is independent of age.
Okay, great. Okay, so are there any further, I don't have any questions in the list, but I'm going to give people another minute in case anybody else is typing or would like to ask another question. We'll just take a pause and give you all an opportunity to make your make another question or a comment. Okay. Well, nobody popped up with any. Did somebody pop up? Ah, we have another question. Okay. You were like, oh, shucks. Nope, you're not going to get out of another question. Okay. The next question is, once you find a significant association between a variant and a metabolite, what is your next step?
Okay, in this case I went, well, in this case, we were, so if I'm interested in a metabolite is associated with a phenotype, and then I find a variant which is associated to the metabolite, then I try to link it back to the phenotype, and then ideally do a Mendelian randomization, which hardly ever works, but that would be the ideal scenario, and then you could prove causality.
Okay. Okay. Great, thank you. Any further questions or comments? We're going to give people just a little bit of time to type in case there's anybody that has a follow-up question or a last. Okay, so nothing else popped up. So I just want to thank you all for your attention and active participation in today's NIH Metabolomics Interest Group webinar. As many of you know, this is a new series that we're starting, and so we're sort of in our pilot phase. And I really sincerely want to thank Dr. Meni for doing the initial and inaugural webinar. This was very exciting, and I'm very pleased that so many of you joined us today and showed such great interest and asked so many questions. I think I'm very pleased with how it went, so I do want to thank everybody who participated. This is very exciting for us, and we will be looking to some other seminars on main campus. We're doing that as well. So, and then another webinar in October, and that should have been part of the listserv. It'll be October, November, and it'll be Dr. Carsten Schur from the Will College of Medicine at Cornell. And so we just want to take this last opportunity to thank our presenter, Dr. Meni, for an excellent presentation, and we would welcome your feedback to inform any future webinars. Most of you who signed up for this know how to get a hold of me because it came from the listserv. So if you do have suggestions for speakers or anything else you'd like to see either this webinar seminar series to do, or if you have any suggestions for the NIH Metabolomics Interest Group, feel free to email me personally. We are sort of revitalizing this group, and I really want to hear from people to see what your needs are. So again, I thank everyone for your attention, for your participation, and especially for Dr. Meni for presenting such an excellent webinar today. So we will see you soon on the listserv and look for announcements for our future webinars and seminars. So thank you very much. We'll see you all later. Bye-bye.